frataxin gene in human cells as well. So they kind of a model of the genetic and epigenetic changes that occur also in humans. And when we treated these animals with one of these molecules, which was called 106, we could sort of normalize the level of frataxin, both at the RNA level here, as you can see in 3 different tissues, and at the protein level, as you can see here in the brain. Histones were acetylated again, back to wild-type levels. And of course, these molecules had no effect on wild-type mice. They did not have the repeat, and therefore no particular epigenetic change in that region. At that time, we were showing that the mice were acutely, because this was a short treatment, they only had 3 injections of this molecule on alternate days, could very well tolerate the treatment. We had no apparent toxicity, and on top of that, this treatment was a sort of correcting a biochemical phenotype that we can see in these animals. So these animals had no particular motor abnormality. The level of frataxin are high enough not to cause any clear pathology, but nevertheless they had some reactive gene expression changes to lower frataxin level that were almost entirely, but largely corrected by this treatment. So this was very encouraging. This has been published some time ago, and then we moved forward in 2 directions. On the one hand, to try and understand something better on the mechanism of action of these drugs, and on the other hand, to try and develop and test new molecules that could be even more effective and possibly have better pharmacological characteristics in order to get these to clinical testing. In terms of mechanism of action, and just let me tell you a few things. This is essentially work from Joel Goresfeld's lab. Let me tell you what the 2 things that seem to be important for an HDAC inhibitor, for an histone and deacetylase inhibitor, in order to be able to increase frataxin level. The first is the specificity of target. There are a lot of different histone deacetylases. This is a table summarizing the most common types, and it seems that this particular one, HDAC3, is really important for this effect. So histone deacetylases that are active on HDAC3 and possibly selectively active on HDAC3 are potentially able to increase frataxin level. Now, with the mechanistic link between HDAC3 and frataxin repression is currently being investigated, but has not yet completely been worked out. So we just know that it's important, but we don't know the exact mechanism. And the other important thing is the kinetics of these molecules. And again, this is from Joel's lab. Basically, the molecules that work, like this inhibitor 106, have what we call slow-on, slow-off kinetics. That if you expose cells to this particular inhibitor, you can see that the amount of acetylated histones slowly increases over a few hours and then slowly decreases in the following hours. And this is accompanied by a significant increase in frataxin protein, as you can see here. While if you use other HDAC inhibitor like this one that is currently used in cancer therapy, that are much more potent as inhibitor of this one, you get a sharp increase in histone acetylation, but which is very transient, very fast, goes up very fast and goes down very fast. And there is no effect on frataxin. So we think that this is combination of HDAC3 specificity and slow-off kinetics that is very important. We have been testing recently a few more molecules. This one, in particular, 2 molecules that are called 136 and 109. And these are the results on primary lymphocytes from patients. As you can see, this molecule called 109 seems to be more effective than the other in primary lymphocytes. And it's able to increase at 10 millimolar concentration of the amounts of frataxin RNA up to 8 or 9 times. That is, I'm almost finished. 8 or 9 times, which is basically levels corresponding to normal individuals, not carriers, really normal non-carrier individuals. Now, this has a series of patients. As you can see, most of them respond pretty well, between an increase between 4 and 9 times. So there are a few who do not respond, but this particular one has very small repeat expansions and almost normal, very high levels of frataxin to begin with. We had a look at the duration of effect. The duration of the effect on RNA is shown, is actually shown here, and here. And as you can see, it goes down relatively rapidly, being back to baseline after 24 hours, but the effect of the protein persists for 24 to 72 hours, as is shown clearly here. 24 hours later, though the RNA is down, the protein is still up. We tested these molecules in these kiki mice, and basically what happens is that the drug is rapidly eliminated, but histone acetylation at the frataxin locus persists with a peak at 24 hours, it persists up to 48 hours, and the frataxin mRNA levels in the mouse, so the animal, we are talking the animal now, are elevated up to 36 hours after a single injection, with frataxin protein levels that persist even longer. So the take-home message here is that these drugs seem to have a much more long duration effect than the persistence of the molecule. The molecule is rapidly eliminated, but the pharmacodynamic effect is much more persistent, and increase the level of frataxin can be found for 2 or 3 days after a single injection, which is promising in the sense that if and when this will be moved to human treatment, the dosage may be probably intermittent in terms of maybe a couple of injections or administrations. We don't know yet whether this will be oral or injected per week, for instance, which would reduce very much the risk of toxicity. So here is basically where we are, and yes, thank you. And the preclinical testing of this molecule is now ongoing, and we hope to move with Repligen Corporation to phase 1 testing in humans sometime early next year. Well, thank you very much for your attention, and these are people who actually did the work that I've shown.
Thank you very much, Massimo. This is has been the first talk in one of the spectacles about the inhibitors of histone deacetylase. So any people have questions? I think it's really very relevant. Specific questions to Massimo. Address for the patients, not for the science.
Yes.
Address to answer questions for patients.
Yes, I must say that unfortunately, because of the train that was full, I have to leave relatively early. So okay. If you can get all the questions now.
Okay. Okay. Tony, Aditya.
Thank you, Massimo. I just have one question about specificity, because I believe that these compounds are going to affect the transcription of other genes in addition to